I want to first start out by thanking the steering committee for inviting me to present my research today. I'm very excited to be here. Today, as Liz mentioned, I'll be talking about interpretable deep learning for biological sequence analysis. So I'll start up with some biological motivation here. Biology tends to rely on high-order dependencies. Take, for instance, this cartoon diagram of a promoter-enhancer complex for gene expression. There are many proteins here shown in a different color, and they're interacting with both the DNA as well as with each other. So if we want to build an accurate mechanistic model for this biological system, then we need to know who the players are, where they're binding, how strongly they're binding to DNA and with each other. Of course, different genes use different sets of promoter-enhancer sequences, so coming up with some analytical function for this general system of gene expression is very challenging. Problems like this exemplify why deep learning is such an attractive approach, because deep learning model is a powerful function approximator that can autonomously learn features. So in principle, a deep learning model can be trained on just the DNA sequences, and it can learn the binding sites of each of these proteins, their locations, their binding strengths, and also higher order dependencies between these binding sites. So I know many of you are familiar with deep learning and neural networks, but for the few of you that might not be, I'll just quickly go over a quick introduction. So deep learning is a computational approach to build hierarchical representations of the data, and artificial neural networks are probably the most popular approach to achieve deep learning. The basic component of a neural network is a neuron. Here, this artificial neuron takes inputs X through these edges, which have weights W, and then processes this with some linear transformation followed by some nonlinear activation. We can create a network of these artificial neurons and organize them into layers. Since we want this network to do interesting computations for us, we need an input layer where we can feed in data. We also need an output layer where we can get the results of the computation. The layers in between are called hidden layers. A neural network like this can relate any inputs to outputs with some approximate function that it learns during training. So, however, their main drawback is that it's not straightforward how to access what function it's learned. For this reason, neural networks like this are largely considered black box models. Nevertheless, we can still do interesting things with these black box models, like classify images of chihuahuas and muffins. Or we can use the same network to classify whether a protein sequence belongs to a given protein family. And deep learning, as you've learned all throughout this semester and the previous years, is that it's really gaining momentum in biological sequence analysis. It can predict transcription factor binding sites when trained on just DNA sequences. It can predict accessible chromatin sites, score mutations in proteins, as you learned last week. It can fold proteins, as you learned a few months ago. And it can even perform dimensionality reduction of single-cell RNA-seq data. Now, while many of these applications are very exciting because they demonstrate that a deep learning method can provide more accurate predictions than previous methods, their main drawback is that it's difficult to understand why they make their predictions. In biology, we really want to understand what our models are learning. This is why we tend to gravitate towards more interpretable, mechanistic models. But in the regime of big, noisy, and high-dimensional biological sequence data, it's not always clear what the signals are and how they vary. So it can be challenging to come up with a mechanistic model that provides state-of-the-art predictions. So there seems to be this obvious trade-off between an easy-to-interpret mechanistic model that provides weaker predictions and a difficult-to-interpret deep learning model that provides more accurate predictions. My research aims to bridge these two approaches. My philosophy is that we should embrace the powerful function approximation capabilities of deep learning to learn the relevant features in these big, noisy biological sequence data sets. Then we can interpret what these deep learning models have learned, and this can provide us with new insights into the underlying biology. And also, it can help us to design more accurate and robust mechanistic models. So in this talk, I'll be focusing on two applications where I've interpreted a high-performing deep learning model, and it's helped us to uncover binding principles of RNA-binding proteins. And in the second part of the talk, I'll show you how it's helped us to design better models for protein contact prediction. So proteins bind to RNA through various modes of interactions. Here, an RNA-binding protein, which I'll be referring to as an RBP, is shown in blue, and RNAs are shown in red here. RBP-RNA interactions can form through a sequence motif on single-stranded RNA, a structural motif like this RNA hairpin here, or double-stranded RNA. There are about 1,400 RBPs identified in humans, but many of these are not well characterized, meaning we don't know who they interact with or where they bind. So the larger question I'm interested in addressing here is, what are the sequence and structure preferences of these RBPs? We can study the binding properties of RBPs with affinity selection experiments. This can be done in vivo with technologies like ClipSeq, or in vitro with microarrays like RNAcompete. In this talk, I'll be focusing on RNAcompete. The basic idea of RNAcompete is that we start with a diverse pool of short RNA sequences and let it interact with a protein of interest. A pull down enriches for the RNA sequences that are bound to the protein, and then we can count how many RNAs are bound versus unbound, and this could provide us with a quantitative binding score for each sequence. There are about 244 RNAcompete experiments that cover over 200 unique RBPs, so it's a very comprehensive data set. And each RNAcompete experiment consists of about 240,000 sequences. So we really need computational methods that can sift through these large numbers of sequences and infer relevant features that can help explain these experimental binding scores. Traditional methods to do this are based on k-mers or position weight matrices. There are also deep learning approaches as well, the most popular one being DeepBind. Now, the details of these methods aren't important for this talk, so just suffice it to say that each of them set hard constraints on the number of features that they can detect, the sizes of the features, and the spatial position, and they do not consider the spatial positions of the features, which is going to turn out to be very important for this data set. So I was curious to see whether we can challenge the assumptions made by these previous methods by designing a neural network that's a more general, flexible feature detector. I call this model residual bind. And residual bind consists of 4 convolutional layers. The first layer learns patterns directly on the sequence, so it learns sequence features. And the deeper layers build upon these features in a hierarchical manner. So we're not limited anymore by the numbers and sizes of the features because partial feature representations learned in the first layer can be combinatorically assembled into whole feature representations in deeper layers. And it also maintains general spatial information of the features. Now, if you're interested in the details, you can certainly hang around after and I'll discuss it with you. Yes?
How does it maintain spatial information?
After you do your convolutional scans, you get a feature map where it shows where the activations of each convolutional filter are. Yeah.
Keep that around.
Yeah, for a little bit. Exactly. So if you're interested, I'd be happy to discuss more of this architecture with you after the talk. And you can also look up this reference here to learn more about the details of this architecture. But I wouldn't say it's important so much for this talk. Here is the comparison of the performance of residual bind versus previous methods. Each dot here is a different RNAcompete experiment. And it represents the Pearson correlation between our model's predictions versus the experimental binding scores on held-out test sequences. So you can clearly see that residual bind performs significantly better than many of these previous methods. So while this is certainly very exciting, one thing that I've learned in dealing with neural networks over the past several years is that they can take shortcuts by exploiting flaws in the data set. So it's crucial that we validate what these networks have actually learned. Fortunately, there has been progress on interpreting these black box models. One such class of methods is called saliency analysis. Saliency analysis identifies parts of the data that are important for the model's prediction. Now, there are many variants of saliency analysis or other attribution methods. But for many of these, the basic idea is to calculate the derivative of the prediction with respect to a given input sequence. What this provides us with is a sensitivity of each nucleotide variant towards that model's prediction. If a nucleotide variant isn't important, it will have a derivative close to 0. If it's a favorable variant, it should have a large positive derivative, and an unfavorable variant will have a large negative derivative. And for simplicity here, I'll just represent these saliency maps as a sequence logo. So in this representation, the heights correspond to the importance of that nucleotide. So using saliency analysis, I really want to understand why our model is performing so much better than these previous methods. Is it learning a new motif that the previous methods were unable to detect? Yes?
So it's a nonlinear map. It matters where you take the derivative. Is there some like average sequence that you use as a base point? No.
Not what I've used here, but there are other methods that I listed that do have some reference.
So why do you not need it?
What?
You're taking the derivative of the.
We're taking
For any given input.
Yes, for any given input, exactly. Yeah. So using saliency analysis, I want to explore why residual binding was performing so much better than previous methods. And for validation purposes, I'm going to show you a detailed exploration for an RNA-compete experiment for RBFOX1. And this is really because it has an experimentally verified motif, this UGC-AUG. Here's a scatter plot of the experimental binding score versus our predicted binding scores on held-out test sequences. Each dot is a different test sequence, and it's colored by the number of mismatches of the best matching subsequence to this canonical motif. So in general, sequences that contain the motif have high binding scores, while sequences that have less of the motif have lower binding scores. But notice these three sequences here that all have high binding scores, but one of them has a mismatch and another has two mismatches. By performing saliency analysis on these three sequences, you can see that a single intact motif is sufficient for a high binding score here, while these other two sequences with mismatches tend to have more instances of their suboptimal motifs. So our network has basically learned that a strong binder is good and a bunch of weak binders is equally good. Now, while saliency analysis is very informative, it does have to be performed on an individual sequence basis. So we have to physically look at these things and deduce what we think the network is learning for ourselves. And this can be very challenging sometimes, especially when these saliency plots are not so interpretable. And I'll show you an example of this a little bit later. So I've been developing a new interpretability method, or I should say an underutilized interpretability method, to test hypotheses that we formulate from saliency analysis using in silico experiments. The basic idea is that we can embed hypotheses patterns inside null model sequences and send them through the network. This provides us with a distribution of the statistical importance of just that pattern while controlling for background noise and other confounding signals that may exist in experimental sequences. So, and if you systematically alter this embedded pattern, you can start teasing out specific functions that the network has learned. So here I embed different numbers of the RBFOX1 motif in random RNA sequences, which turns out to be a good null model for the RNA-compete dataset. And you can see that residual bind learns an additive model for each additional motif here. While it's clearly linear here, other RBPs may be nonlinear, so it could be a way to test whether your network has learned properties, nonlinear properties like cooperativity. And from the saliency plots, I also noticed that sometimes there are these seemingly overlapping motifs. This UGC-AUG overlapped with the UGC-AUG. When I saw this, I thought, if one of these sites is occupied, it could potentially sterically block the other site from binding. So I would guess that this overlapping motif should have a lower binding score compared to a sequence with two binding sites that are well separated. But this overlapping motif should still have a higher binding score compared to a sequence with just one site because there still are more chances for binding. By designing synthetic sequences with these patterns, you can see that residual bind indeed learns a function that's consistent with our biophysical expectations of steric hindrance. So before I continue, I want to emphasize 2 points here. The first point is that what I've essentially showed you here is that we can treat a trained neural network as an in silico laboratory where we can perform in silico experiments for free. And this is, of course, in lieu of actual wet lab biological experiments that can be time-consuming and costly. The second point I want to make is that saliency analysis is really technology borrowed from computer vision. But this treating these in silico experiments approach is really specific, I think, to biological sequence analysis. And that's because it's relatively straightforward for us to systematically design sequences in genomics, but it's not so clear how to do so for natural language processing and computer vision. So one major factor that we have not considered so far is RNA secondary structure, which can certainly influence the binding affinity of a motif. So previous methods based on k-mers and position weight matrices have found that including secondary structure predictions directly into their model as input features significantly boost their model's accuracy. So I was curious to see whether it could help residual bind. So like the previous methods, I used RNA-PL fold to calculate the secondary structure profiles. And what RNA-PL fold provides you with is a probability that each nucleotide is paired or unpaired. So I retrained residual bind, this time using this paired-unpaired secondary structure predictions as an additional nucleotide channel. Yes? Oh, OK. But we didn't notice any significant improvement across all the RNA-compete data set. Here's a box plot of residual bind trained on just sequences. And here we have residual bind trained with additional paired-unpaired secondary structure predictions, which I've labeled PU. This was surprising because we know there are RBPs in this data set that have structural preferences, such as VTS1, which preferentially binds to this motif in the context of a hairpin loop. So using saliency analysis, we can try to explore what's going on here. And we performed saliency analysis on 2 sequences with a high binding score, and we found that this VTS1 motif is clearly present here at the center of the sequence. But there are other nucleotides that are important as well, and it's not really clear what these other nucleotides represent because there is no consensus. So I performed saliency analysis, this time using our model trained with secondary structure predictions as input into the model. So here the U's and P's underneath the sequence represent the unpaired and paired probabilities that were input features to the model. And below that are the saliencies for those secondary structures. Now you can see that this sequence motif is much cleaner, and it's located in the unpaired region flanked by paired nucleotides on either side, which represents a hairpin loop, as we would expect for VTS1. I also performed saliency analysis on sequences that contained the VTS1 motif but that had lower binding scores to see what's going on here. We notice a similar trend where you can clearly see that the motif is present here, and there are lots of nucleotides that are important as well. But when we include the secondary structure predictions, you can now see that this sequence motif is located in the paired region of the hairpin. So the fact that our model's performance doesn't improve whether or not we include secondary structure predictions into our model suggests that it may actually be learning these secondary structures directly from the sequence. And these other nucleotides that it finds that are important might be signatures of those secondary structures. And this was actually surprising to us at first because we're using a convolutional network here with these big, bulky convolutional filters, and it's unclear how it can pinpoint exact positions and learn higher order base pair interactions. But to test whether our network can actually learn secondary structures from sequence, we designed a simpler synthetic data set that consists of RNA sequences that contain a hairpin versus random RNAs. And here, the only discriminating feature are the Watson-Crick base pairs that are in the stem here. So after we trained residual bind on to classify whether a sequence contains a hairpin or not, it was able to do so quite easily. Its performance was nearly perfect on held-out test data. But as I mentioned before, it's crucial that we validate what these networks have learned. So we started out by performing saliency analysis on a sequence that contains a hairpin loop. And you can see that the nucleotides that it finds are important fall right in the stem region of this hairpin. And the stem region is annotated in black underneath the sequence. While this is consistent with what we'd expect, it still doesn't prove whether residual bind is learning a physical model of base pair interactions between specific positions. And we fundamentally can't get this information from saliency analysis because it's a first-order interpretability approach. So with the help of Stefan Paul, an undergraduate researcher who's now graduating tomorrow at Harvard, we developed a new we developed a second-order interpretability method that we call second-order in silico mutagenesis. The basic idea is that for a given sequence, we're going to take two positions and generate new sequences with all possible pairwise mutations while keeping the rest of the sequence intact. And we're going to send those sequences through the network to get out the predictions. We can organize those predictions as like this into a 4x4 matrix for positions 1 and 2, as you see here. Now, if those two positions are directly base paired, we would expect that all complementary nucleotides should maintain that base pair, and so the model's prediction shouldn't change. That's represented as white here in this off diagonal. While any other combinations of mutations should break that base pair, leading to a decrease in the prediction, which is represented by a darker color. So we perform second order in silico mutagenesis for positions 6 through 16 against positions 24 to 34. And you can see that this Watson-Crick base pair pattern is really found only along the diagonal here, which are precisely the positions that we would expect to be base paired with each other, as shown by this diagram here. So this really proves that residual mind is learning a physical model of base pair interactions between specific positions. Now, you probably noticed that there is some funny business happening around each of these corners here. And this turns out to arise because convolutional filters have these big, bulky filters. So they have problems defining the edges of these stems. So Stefan Paul has since gone on to explore different kinds of neural network architectures and see how well they can learn more complicated RNA secondary structures on actual biological sequences. And if you're interested in learning more about that, I'd be happy to discuss it with you after the talk. Now, one final point I want to, one interesting observation I want to make is that for a lot of the RBPs in this RNA-compete data set, I noticed that saliency analysis on the top scoring sequences had this weird little GC content on the 3' end, on the right side here. Now, when you see this across so many different RNA-compete experiments, it's unclear what the effect size is or whether it's even that important or not. But now that we can treat these neural networks as in silico laboratories and perform in silico experiments, we can try to measure the effect size. So what I did here is I just sent random RNA sequences through, and then random RNA sequences with GC content on the 3' end, random RNA sequences with the motif at the center, this known motif, and then the motif at the center with GC content on the 3' end, and you can see that the predictions increase significantly. But when I move this GC content from the 3' end to the 5' end, the left side, the effect is lost altogether. Because I noticed this across so many 170 out of the 244 experiments, this really doesn't, I don't think it's a natural property of RNA binding so much as it is a bias with this experimental assay. So, so the takeaway is that we can, even though we wouldn't expect this GC content to generalize, it's still a powerful tool, a data science tool to help us understand the signals in the data. Now I'll go on to the second part of my talk, where I'll show you how interpreting a deep learning model has helped us to design better models for protein contact prediction. Yes?
Yeah, so before you do that, I mean, is it necessarily surprising that you see that your network can learn, for instance, secondary structure, right? I mean,
yeah.
that doesn't seem very surprising, right? So
it's not surprising, but it hasn't been shown that these networks can learn secondary structure yet. So this is the first demonstration, to my knowledge, that that people have actually shown that.
I guess my point is that
it's always assumed, I would say.
Well, like for instance, if you take an example from computer vision, right? You find that the earliest convolutional layers will do rudimentary edge detection, much like what you see in the human visual cortex.
Yes.
But there's no guarantee necessarily that that's going to be the case. And in fact, you have to constrain the number of filters, at least for the deeper layers, in order to get that sort of emergent structure. So I guess my question or challenge to you is, I mean, it's nice that one can perform in silico experiments, if you will, on your model in whatever learned state that it has, but I mean, if you go fishing long enough, you can always find things that, as a biologist, we can rationalize are biologically meaningful.
Yes.
And those things that we can't rationalize, well, we just sort of put those off to the side. So, I mean, what new stuff do we learn from this sort of approach?
I guess what we've learned in this case, I was going to highlight that later on in the summary slide, but we've learned why these models are doing better than those previous methods because it's able to integrate the number of motifs and suboptimal motifs, unlike a PWM approach or even something like deep bind, which max pools across the entire sequence, losing all spatial information. So it's helping you understand the relevant signals in your data and even learning that GC content on a 3' end, not the 5' end or any other parts of the sequence significantly matter. So it's like now you can go back and redesign your mechanistic models in considering these signals that you've learned using this deep learning approach.
Maybe you didn't show this, but did you perform the same exact type of saliency mapping and the in silico modeling with the other say neural network-based models to show that they don't have these recoverable features?
No, I did not, but they max pool across the entire sequence, so you essentially can't get that information. You lose that spatial information.
But you may be able to at least go in the initial convolutional layers, right, and see what sort of...
Yeah, but I just showed you in the previous slide, GC content on 3' end versus 5' end is significantly different, and if you max pool across the whole thing, it doesn't matter. So now moving on, in December of last year, DeepMind announced that their model AlphaFold was the winner of the Cas13 protein folding competition, which is certainly a big win for the AI field. This was surprising because I remember when they first announced that they were going to address this problem, a lot of scientists I know thought that there was no way outsiders without any domain knowledge would be able to make a significant impact in this field. And to an extent, they're still kind of right because some of the people that worked on this project had protein folding backgrounds. But I say this mainly because AlphaFold is not an AI system that learns how to fold proteins all on its own. It actually uses a lot of domain knowledge. And the main input feature they use is the convolution in the amino acids in proteins. So in proteins, we like to think that positions that are evolutionarily conserved tend to be are functional. And we can assess conservation with a multiple sequence alignment, as I have here, where we have homologous, the same homologous proteins across different species aligned together. Now, positions that are conserved tend to have the same amino acids across species, like this column in green. But two positions that are directly interacting with each other to maintain a structural contact do not necessarily need to be conserved, so long as they can make compensatory mutations, like these two positions that are covariant in blue and yellow. In this cartoon diagram, you can see that this protein sequence has their amino acids swapped here, but they still effectively have the same structure. Convolution signals like this are actually more prevalent in RNA secondary structures. In proteins, we believe it's a little bit more nuanced. Our current working hypothesis is that amino acids evolve in a more incremental manner. For example, here I have a protein that only requires one contact to maintain this structural fold. If a mutation were to occur where it now forms two contacts, well, this becomes redundant. So one of these positions can now mutate without affecting the organism's fitness. And so what this means is that convolution signals in proteins are not necessarily compensatory, but they may still exist to some degree because we believe that evolution is constantly subsampling these contacts. So what this means is that we can take a multiple sequence alignment and still infer protein contacts by measuring convolution. Although the signals aren't as strong as RNAs, they're still present. So here is an example of a contact map where they actually show the position of the protein from the N terminus to the C terminus going in both directions. The gray dots represent the known contacts from a known structure. And the blue dots represent a convolution predictions made by a model I'll describe in the next slide. And in general, you can see that the convolution predictions overlap nicely with the known contacts. So how do we measure convolution? Two popular ways of doing this are mutual information and a POTS model. Mutual information measures the overlap in information between two positions. So a high mutual information indicates that the amino acids in those two positions are highly coupled, while low mutual information indicates that they're more or less random. A Potts model comes from statistical physics, but it's known in other fields as a Markov random field or an undirected graphical model. We use a Potts model to learn the probability distribution of a multiple sequence alignment. So a Potts model contains one-body terms, these H's here, that learn site-independent conservations, and two-body interaction terms that learn all possible pairwise covariation between all possible position pairs. And you've learned about this with Sergey's primer, as well as Debbie mentioned it last week as well. Now, before I continue, I want to emphasize that Potts models are currently state-of-the-art at predicting protein contacts directly from sequences. And also, the main input features by AlphaFold are the parameters of a trained Potts model. Now, before I continue, I want to emphasize another point, that our mutual information and a Potts model only consider dependencies between positions. But there are also dependencies across sequences, which arises due to the phylogenetic relationship of the organisms. So our models can't distinguish covariation between these two columns and covariation that aren't really covarying, but that are just phylogenetically inherited mutations, such as these two columns here. Because it's unable to distinguish these two events from each other because they don't consider phylogenetic information, their contact predictions are typically fraught with a lot of strong false positives. So here is an example of a protein contact prediction made by a Potts model. You can see that they have a lot of these strong plaid patterns here, which don't correspond to any contacts. But there are heuristic corrections that can reduce or remove a lot of these plaid patterns to get more accurate contact predictions. So just to reiterate, our current models are fundamentally flawed because they do not consider phylogenetic signals, so we have to make heuristic corrections to their predictions. Now, excuse me, while a Potts model considers one-body and two-body interactions, proteins can have higher order interactions. So it's intriguing to consider a neural network approach because there aren't any strong restrictions on what they can learn. A variational autoencoder is one promising approach, and I'll be referring to these as VAEs. A VAE learns a distribution of the data, and it does so through an approximation called the variational lower bound. A VAE solves this by approximating each of these distributions with a neural network. So a VAE consists of two modules. The first module uses the neural network to approximate this conditional distribution Q of z given x, where z are the latent variables of the model. Then a second neural network approximates this conditional distribution P of x given z, which this network can generate data given latent variables. Now, both of these networks are trained simultaneously by maximizing this variational lower bound in an unsupervised manner. And it turns out that these latent variables learn to capture variations in the data. Here, I've replicated results from Kingma et al., where we've trained a VAE on the Frey face dataset. This consists of Brendan Frey making different kinds of facial expressions. And what I've done to generate this plot is I use a second module of this VAE, the second neural network here, and I systematically generated data from different latent coordinates to map out a manifold of what this VAE has learned. And you can see that one latent variable has captured his pose looking left and right, and another latent variable has more or less captured his facial expression, smiling, frowning. Now, in collaboration with Sergey, who's the lead developer of a very popular Potts model called Gremlin, we set out on a large-scale exploration to see how well these VAEs can learn contacts. Here is one example for a histidine kinase. Here are contact map predictions, or here are known contacts from the PDB database. And here are contact predictions made by mutual information and by a Potts model. And these two are, of course, after heuristic corrections. In general, you can see that the Potts model visually matches these known contacts much better, while mutual information has a lot of these strong false positives that seem to drown out the dimmer contacts that are underneath. And this is a well-documented problem with mutual information. Now that we've come to our VAEs, it's not actually clear how we extract out contacts from them, from a trained model. But we figured that since the VAE is essentially performing dimensionality reduction through a small number of latent variables, analyzing the generated sequences might provide a cleaner, denoised representation of the contacts. So we calculated MI on the VAE-generated sequences, and we're surprised to find that it doesn't seem to be capturing a lot of these contacts, but it does seem to be capturing the false positive signals quite well. So we were confused at the time, but we said, what the hey, why not just subtract this MI observed minus this MI VAE generated? This is what we call MI VAE corrected. Now visually, you can see that the contact predictions seem to visually match these known contacts much better. And in the field, we can quantify these contact predictions. It's typical in the field to calculate the fraction correctly predicted versus the number of predicted contacts. So what I did to generate this plot is I took a lower triangle of one of these contact maps and I sorted it in descending order. Then I systematically went through the top X number of contacts and I calculated what fraction of those matched the known contacts. And mutual information is shown in orange here. MI on the VAE-generated sequences is shown in red. Potts model is at the top in blue. And MI with this VAE correction is at the top right in brown, right next to the Potts model. So while we're disappointed that this VAE doesn't seem to be capturing contacts, it does seem to be learning some an excellent background, a null model for mutual information. Now comes the question of what is the VAE learning and what makes a good null model? So to interpret a VAE, it's typical to see how the latent variables organize the data. So I trained a VAE model with two latent variables, and this is the latent space that you see. Each dot represents a different sequence in the multiple sequence alignment and is colored by the organism's phylogenetic class. Here, I'm only coloring the top 5 most populated classes, and everything else is grouped into this other category in blue. In general, it looks as though these latent variables are clustering sequences according to their phylogeny at the class level. And this is an observation that was also made by the Marks lab with their VAEs. Now, another way we can interpret these VAEs is by looking at the generated data. But we're dealing with protein sequences here, and they're not as human interpretable as images of faces. So I'm only going to highlight some of the main results here. Now, the second module of that VAE, this conditional distribution P of x given z, maps latent variables to a sequence model. This is the probability of each amino acid at each position, something that I'll call a profile. So each position in this latent space maps to a unique profile. And so by comparing the profiles generated by different latent coordinates, what I found is that sequences within a cluster generate very similar profiles, while sequences across different clusters generate very different profiles. So it seems as though this latent space has this star shape because the VAE is trying to maximally separate out each of these clusters, which are each defined by their own unique profile. Now, I also did further explorations on different kinds of VAE architectures, using different kinds of latent variables, and also explored synthetic data where we simulated sequences down a phylogenetic tree with structural couplings. And they all support these same findings. So taken together, what we hypothesize this VAE is actually learning is a profile mixture model that's capturing phylogenetic signals at the class level. So a profile mixture model, what it does is it essentially groups together sequences that are similar to it and generates a profile model for each group cluster of sequences. So importantly, a profile is a site-independent model. So it cannot capture structural couplings by design. So what this means is that sequences generated by a profile mixture model should only contain phylogenetic information and not any structural information if we believe that structural couplings and phylogenetic couplings are distinct. And that's what we believe. So of course, it makes sense that sequences generated by a profile mixture model should serve as a good null model because they only contain phylogenetic information, whereas our VAEs and POTS models and mutual information are learning structural information as well as phylogenetic information. So this is just, of course, this is just a hypothesis. So to test whether our network can actually learn, whether a profile mixture model actually learns, our VAE actually approximates a profile mixture model, I designed a profile mixture model in a Bayesian framework with variational inference. And I have a slide that discusses all the mathematical details of this approach. But I'm not going to discuss the details right now. If you're happy, I'd be more than happy to discuss it with you after the talk. But in general, after you solve this, this variational inference problem, you end up getting something that looks like a expectation maximization algorithm. And importantly, we use Dirichlet priors, so it prunes down the number of mixture components to a suitable number at the end, similar to what the VAE does. So we call this model a Bayesian Profile Mixtures, or BPM. And we find that it performs similar to this VAE. So here we have MI on the VAE-generated sequences and MI on the BPM-generated sequences here. And here we have MI with the VAE correction and MI with the BPM correction. Both are visually very similar, and their performance is also very similar, as shown here, where their curves are in red and purple at the top right next to the POTS model. So this provides further evidence that our VAE is actually approximating Bayesian profile mixtures, Bayesian profile mixture model. And I find this to be pretty remarkable that by interpreting this VAE, we're able to come up with an analytical function that can replicate its performance. And moving forward, a major benefit of this BPM is that it's less susceptible to overfitting compared to this VAE. And if the VAE isn't regularized heavily enough, it can essentially memorize most of the sequences in the multiple sequence alignment. And if you do that, of course, you're going to get structural information on the generated sequences, by the way. And now that we know our profile mixture model can capture phylogenetic information, we have clues on how we can fix our current POTS model by extending the one-body term to a mixture of one-body terms to absorb the phylogenetic signals into that rather than being learned in the two-body interaction terms. And another benefit is that mutual information with this Bayesian profile mixture correction is, while it doesn't perform better than a POTS model, it's significantly faster to train than a POTS model. So we can now begin addressing problems that are currently prohibitively too slow with a POTS model. So in summary, what I've showed you here today is that deep learning is not just a black box predictive tool. We can interpret a deep learning model, and it can provide new biological insights and help us to design better models for protein contact prediction. In the first part of the talk, I showed you that deep learning is a powerful tool to learn sequence and structure motifs. I use saliency analysis and in silico experiments to help us interpret a trained deep learning model. And we found that our model considers not only finding the motif, but the number of motifs and their spatial positions. And it also learns structural preferences or accessibility of the motifs if it's important for that RBP. And of course, it has to be very flexible because these properties vary from RBP to RBP. And in the second part of the talk, I showed you that VAEs can help us understand dependencies in homologous protein sequences. And ways we can interpret the VAE is by not only exploring how the latent variables organize the data, but we can also analyze the generated data with traditional tools, which helped us to interpret this VAE. And we found that, that our VAE, when it's trained on a multiple sequence alignment, learns a mixture model, a profile mixture model, which captures phylogenetic signals at the class level. And this turns out to be a great null model for mutual information. And we also have clues on how to fix our current POTS models. Now, with that, I'd like to thank my advisor, Sean Eddy, and my collaborators, Stefan Paul and Praveen Anand, who were collaborators in the first part of the story, and Sergey, who was instrumental in the second part of the story with the VAEs. Thank you.
Have you considered other null models for mutual information? The simplest one is normalizing, let's say, by minimum entropy, but then others, like, for example, I've developed one a long time ago that's very popular in the network space context, like the relatedness, that basically normalizes for the distribution of mutual information. It's a slightly different context, but in other words, have you tried normalizing mutual information in other ways? It's known to be biased, it will be biased.
Yes.
It's a common problem that is pervasive to any use of mutual information.
I'd be happy to discuss it in detail with you after.
So in the first part of your talk, you mentioned how you trained a network that identified a submotif that you thought was just like a bias in the assay that generated your training data. I was wondering if you have any advice for how you would use models where you kind of know that they're likely to learn a bias like that?
Yes, obviously if you have a model like that, you wouldn't expect it to generalize on in vivo sequences which don't have that specific bias, right? And so you really, this is why it's really important to go beyond just training these models and treating them like black boxes and then just saying, I'm going to generalize it on this and it generalizes slightly better or something. You really need to interpret these models so that you can understand these signals. So the way I envision how this can help us is, well, we can, either you mask that signal during training, or what I'm trying to do is to figure out what the important signals are and how we can adjust our current mechanistic models because they're so much more reliable right now. And to make sure that these effect sizes are real, we can include just the GC content on the right side into our mechanistic models and just on the RNA-compete data set and show that its performance replicates the neural network. And then you take off that component when you're trying to do your generalization task.
I have a question. So back in the days of RNA secondary structure prediction, I had some folk wisdom that I carried with me, but I don't know if it was true or not. And now I especially don't know if it was true. But we always said that the secondary structure prediction models would give you like a top 10 list.
Yes.
That the first one wasn't necessarily right, but it would absolutely exist and somewhere in that top 10 list. Choose one, not necessarily ordered even. If you had fed that hard information that it had to correspond to one of these predictions, but not necessarily just the top one, would that have given any information? Would it have been?
So that was in terms of, are you referencing how I was including the secondary structures into the model, right? So that's precisely the problem, that if you just use a structure prediction tool that gives you one structure, it's likely not to be correct. So what RNA-PLFold does is it subsamples a lot of suboptimal structures and it averages the probability that each nucleotide was paired across all of those suboptimal structures to give us more of a probability rather than saying that this is the structure.
So it softens the notion of
Exactly. Because if you're going to make it hard, then it's likely to be wrong.
within it, so it's hard to search.
Yes.
For the second part of your talk, so the variational autoencoder kind of trying to maximize the amount of variance it explains, but just in a nonlinear way, right? Mm-hmm. So I might just regard it as a kind of a nonlinear PCA in a sense. And then when you find it removes the amount of phylogenetic causes, it might be just the phylogenetics are the top two kind of.
Absolutely. Absolutely. So there has been a study that's looked at PCA on the covariance of these multiple sequence alignments, and by removing the first few principal components, you can actually recover structural covariations after. And that's because if you remove the first few principal components, those are the highest varying ones, those are all containing phylogenetic information. And I had a reference for that earlier when I had that slide up. So,
then
yes.
my question is, why does the Potts model actually can't consider, can't have the correct prediction? Is it because they already accounted for the, phylogenetic reasons, or?
Sergey, when he gave his primer a few months ago, he gave his explanation for why he believes that the Potts model is indirectly handling phylogenetic signals, and the main reason that our current working Sergey's current working hypothesis, I'm sorry if I'm overstepping my bounds because he's right here, is that the phylogenetic signals are all coupled with each other, whereas structural contacts are coupled with just, structural contacts are just coupled with each other, so you only have two contacts that are coupled, whereas phylogenetic signals, you have a lot of positions that are all indirectly coupled with each other. And so the way this Potts model tries to give contributions to this using L2 norm is to just downweight all of those. And that's sort of our current explanation for why the Potts models still work.
All right, let us thank.
I don't know where it's coming from. Oh, okay.
So in your last slide, you said that, at the top, that your approaches enables discovery. You found new biological insights. So what new biological insights did you find?
I guess it's not necessarily a biological insight, but it's a modeling insight that the current models are not considering the spatial information, which is important, I would say. And some of these models aren't considering that you have to integrate the number of motifs and suboptimal motifs throughout the sequence, and that affects these binding scores.
Do you see a path to maybe if you sort of recapitulate a lot of the sequence features that we know to be important, like secondary structure that you had previously captured in predictive models, that you could potentially interrogate the models, guess new features of interest, and then validate them?
Yes.
In vitro experiments?
Yeah, that would be amazing. That's the line of research where I want to head down towards. So I'm working in this field where it's more well-defined, and so that we can actually understand these models first. And now I think we have enough tools that we're able to actually go out and make bold claims of new biology by working on more complex problems.